Let's spend some time talking about PCR. This is sort of a revolutionary technique that's completely changed the world of DNA analysis. This is this multiplication technique we talked about. It stands for polymerase chain reaction testing. And what it is, and of course you may have heard of it with COVID testing. They talk about having a PCR test as being one example. So they take a sample and they try to do a PCR and amplify it so there's enough so they can do a really good version of testing on it. So we use this in multiple applications, but specifically in DNA, it's a process by which I can make multiple copies. So I'm going to replicate small quantities of broken pieces of DNA. I can only do it for short little lengths. I can't do it for this whole thing. But I can basically use this. Polymerase is a natural enzyme in the body that is the thing that does the replication in our cells. But we kind of have a specific way of doing it where we can make many, many copies of short segment. So it amplifies these tiny samples into many million copies. And now, if I have many million identical copies, I can do great analysis on them. So this is the procedure. First, I have to take my DNA and I have to separate it. I have to heat it and separate it into single strands. By the way, this DNA would already have to be a short segment. So remember, this is not the entire DNA. It's not the entire chromosome. It's not even the entire gene. It's a short segment that has already been taken out of the full DNA. I take it, I heat it, it separates into the single strands when I heat it. Then I have to use these things called primers. So this has to be, I know what segment of DNA I have, and I have these special primers or short strands of DNA that can bond in a complementary way to the thing I'm trying to analyze. And then once I can do that, the polymerase is added and it starts making copies. So now I'm going to use this power for good and I'm going to make multiple copies on it. I also have to add the free nucleotide so that it needs to have base pairs to add on. So I let this, so I do this once. So I heat it up, put the primer in there, it attaches, I add in the polymerase and the base pairs, it starts making copies, and then it stops. I cycle it again. I heat it up, break it apart, I put in more primer, more polymerase, it makes more copies. So I can repeat that 25 to 30 times. Each time it's making a duplicate of what I have. So it's like it doubles the quantity. Then on the second time, it doubles it again. So that's now at quadruple. It doubles it again. It's now at times 8. Doubles it again, 16. Doubles it again. So each time, by the time I've done that 25, 30 times, I have now multiplied all of my times so many times that I have multiple thousands and millions of copies. So within a few short hours, I have basically a billion-fold increase. So this is kind of looking at what we're doing in a visual way. Again, here's my short segment of DNA. First, I have to denature it and anneal primers. Basically what that means is first I heat it up and I unzip it so I get 2 single strands. And then I anneal. Anneal is meaning like I'm sticking the primers to it. So these yellow and pink things are the primers. So they stick to these segments of the DNA that I have separated out. Notice now I have 2 strands instead of this double strand. I add in the polymerase and the free bases, and I do a run. And now I make 2 copies. So now I made this new blue copy. So now I've done a full duplication. Now I take that, I repeat the process. I denature it, I add more primers, and now each one of these can make another copy. So the first copy, the original copy was the blue, the first copy was the dark blue, and now I'm making the green copies. So now I have 4 copies. I do it again. In this case, I'm separating them all. First I'd heat it up, separate them all, add on new primers, and now it's making the beige repeats. By the time I've done that, I have 8. I do that 20 to 30 more times. That's how I get my millions of copies. This is huge. This has revolutionized DNA work because now, trace DNA found under fingernails, decayed bodies that normally it degrades the DNA and you can't, but I only need small segments of it. I can amplify it up. So at this point now, DNA work has become super precise. I can use very minute quantities to get some data analysis off of it.